Greetings, students, and welcome back to part 2 of our unit on viruses and prokaryotes. Today we're going to talk about prokaryotes, specifically eubacteria and archaebacteria. There was a time, not too long ago in recent history, I guess it was about 30 years ago, they changed it or so, that bacteria used to be lumped together in one kingdom. Archaebacteria and eubacteria used to be lumped into a kingdom that we called Monera. But recent developments in technology and scientific equipment and our understanding of these organisms have made it such that scientists felt now today they have to separate them into two different groups. So if you look at the picture to the right, you'll see these two specimens here of bacterium. Both of them look very, very similar, don't they? However, on the left you have eubacteria and on the right you have archaebacteria. And what scientists have found recently is that these guys are so different at the molecular and chemical levels that they no longer can be classified together in the same group. So now we have two distinct groups. We have the eubacteria and the archaebacteria. Eubacteria are your modern day bacteria, found just about everywhere. And then the ancient bacteria, the ones that have been around since the beginning of time, are the archaebacteria. In fact, the name archaea actually literally means ancient. These guys can live in just the most inhospitable environments. They can live in harsh environments like the methanogens that produce methane gas as a byproduct. The halophiles, which love excessive amounts of salt and can live in places where others cannot live. And then the thermophiles that love extreme temperatures that would like pasteurize normal bacteria. These guys think of it as like a sauna or a hot tub, and they sure do enjoy it. But for a long time they were classified together, but no more. Today they're in two distinct groups. So how are bacteria, primarily eubacteria, classified? Bacteria are classified by four different things: by their shape, their cell structure, the way they move, and their metabolic processes. Now there are three main shapes of bacterium. There are more than that, but there are three primary ones. They're coccus, which are round bacterium, like these guys here, spherical in shape; spirilla, which are these spiral-shaped ones; and then bacillus, which are these rod-shaped ones. There are other shapes as well, like vibrio, and some that fall a little bit in between, but these are the primary distinct main lines of shapes in bacteria. And some of these you'll even find in the scientific names of some of the bacterium that we'll be looking at in the class on the microviewers and in the projects we'll be doing. Primary structures of a typical typical bacterium include the cell wall on the outside. And remember the case of like eubacteria, it's made of peptidoglycan, a cell membrane underneath, DNA, which is this stuff found right here, sometimes referred to as a plasmid. Remember, it's not like a traditional eukaryote. It's not membrane bound in a nucleus. It's found inside the cytoplasm. Ribosomes for making proteins, flagellum for movement, and then pili right here, which allow bacterium to attach to surfaces to help facilitate movement, and also facilitates the process of conjugation, which is a form of quasi-sexual reproduction that takes place in bacteria. Some other characteristics of bacteria include describing the way they get their food. Some are autotrophic, like the cyanobacteria. Some are heterotrophic, enjoy eating stuff. Some prefer to be in oxygen-rich environments. Those are called obligate aerobes. Some like to be in non-oxygenated environments. Those are obligate anaerobes. And then some don't really care if they're in either type of environment. Those are called facultative anaerobes. They can live either with or without oxygen. Now, how do bacteria reproduce? The primary method of reproduction in bacteria is a process called binary fission. Binary fission is a form of asexual reproduction, in that the parent bacterium will divide and produce two identical copies. A little bit like mitosis, remember what you learned about earlier, but of course remember there's no nucleus to divide in this case. And it could happen very quickly. Look at this, some could divide as rapidly as every 20 minutes. Then there's a process called conjugation, in which two bacteria hook up together with the pili, and then there's an exchange of DNA. I'll show you that in the next slide. And then something cool that bacteria can do is they can produce what are called endospores. Endospores are protective coatings around the bacterium that allow it to stay dormant for months or even years in conditions that are unfavorable. And then when conditions do become favorable, the bacterium can become live and active again and start reproducing and stuff. In the bioterrorist attack of 2001, in the United States when they were mailing anthrax around the nation. The anthrax that was contained in the envelopes were in spore form. And then when they were inhaled by the unlucky people that actually opened the envelopes up, the anthrax would go into their respiratory tract and then become active. Here's a slide on binary fission. You can see here the process starts here with the parent cell. And then the cell will begin to divide up the plasma membrane. A wall will form to divide the two cells into separate pieces, with each having their own identical pieces of DNA, and then you end up with the two daughter cells, which if you look at the image here, you can see, of course it's just an artistic image, but you can see the identical DNA in each one. So this is an example of asexual reproduction. Contrast that with conjugation, which is a little bit different, because conjugation, as described in this slide here, is a parasexual process, because you do have some exchange of genetic information. Look, some of this DNA is being sent through the pilus to the other side, in which it's going to be incorporated into the DNA of the cell that's receiving the information. Once that occurs, then this cell can divide through binary fission with that alteration in its DNA, and now the daughter cells will be different than the parent was before it received the DNA. Now, I don't want to spend too much time emphasizing it because we'll have our projects on it, and it's really not a fair way to describe bacteria, but there are some bacteria that are bad. They can make us sick by harming our cells or producing toxins or poisons, like botulism in a damaged can. You can see some examples of some nasty bacterial infections here. Strep throat, staph infection over here, ouch, or these cavities here. Now, most bacteria, on the other hand, though, are inconsequential to us or harmless. They're all around us. They don't bother us. They're, like I said, inconsequential or harmless, and some are actually even beneficial, and we're dependent on them. For example, bacteria are decomposers, which means they break down things like dead and rotting material and recycle that stuff back into the soil, which is very important to keep the soil so that it can be used to grow new plants. Some are nitrogen fixers. Now remember, plants love nitrogen. It's one of the three main things they love, aside from phosphorus and potassium. But the problem is nitrogen is found primarily in the air. It's like 78% of the composition of our air, but plants can't extract it in form that's gaseous like that. So they live in a symbiotic relationship here. You'll see in these soybean root systems here, you see these nodules contain the bacterium rhizobium, which can actually capture that nitrogen and turn it into a form that plants can use. This is a process called nitrogen fixing. We also have E. coli in our intestines that help us break down our food and help produce vitamins. Now, E. coli can be dangerous in other places, but in our intestinal and digestive tracts, it's very beneficial to us. They are important in the production of foods, for example, like yogurt. Scientists use them in genetic engineering and in research they do with new medications and stuff. How do you treat bad bacteria, though? Let's get back to that in the previous two slides over. Some bacteria can be treated by vaccine, similar to the way they deal with the virus. Great example is anthrax. They have a vaccination for that, Bacillus anthracis. That vaccine is not generally given to the population, for most of us will never be exposed to anthrax, but they do provide it to soldiers and stuff, and like maybe zoo workers and things like that that might have a higher chance of being exposed to anthrax. So there are some vaccinations for bacterial infections listed here. The primary method of treatment of bacterial infections is by antibiotics, which are really wonder drugs when you think about it, because they're designed to be ingested and kill something living without harming the host organism that's receiving the antibiotic. The earliest antibiotics were developed by Alexander Fleming in the early part of the 20th century. He developed penicillin first, which is a derivative of the Penicillium fungus, which is very effective in killing bacteria. And then, of course, that opened up a whole variety of different types of antibiotics that were developed later. The problem with antibiotics is some of the bacteria, through the process of evolution, we can witness it ourselves here, have become resistant. We learned about that in a couple chapters ago. And some of these antibiotics don't work anymore. Like, for example, the bacterial infection MRSA is resistant to just about everything. Sometimes you just want to clean up a surface that has bacteria on it. You would use a disinfectant for that. Disinfectants are designed to kill bacteria, but not designed to be ingested or usually get on the skin for contact. We can sterilize things that might be exposed to bacterium by heating them, like your mom probably did when you had a splinter. She probably heated the needle under a flame or something. We can take our liquids like milk and juices and stuff and pasteurize them. We talked about that earlier, heating them to a sufficient temperature to kill the bacteria and then vacuum sealing them. And then, of course, we can also store our foods and drinks in the refrigerator, which slows down the rate of reproduction of the bacteria, allowing foods and drinks to last much longer than they would have in the old days. So this slide here concludes the unit. Don't forget to make sure you look over the whole presentation, make sure you do all the readings, look over all the homeworks we did, and the study guide in preparation for our test on Tuesday.